Hello, everybody. It's a pleasure to be here and a pleasure to kind of present what we have been doing and kind of the vision for the next years. We really want to everybody here to feel that genomes were just the start, and although a lot of the slides will involve genomic assemblies and the qualities of assemblies, what we really want to talk about are the next step. So what can research do with many genomes? And I'll follow up on that as I go forward. So what we really want to do is understand genomic diversity for many reasons. And the idea is that genomic diversity is best revealed if you have the full genomes of many, many lines, and then you compare them all to one another. And then you should do something that's meaningful with the results. And what we do is we do very accurate and very high-resolution haplotyping, and then out of that, we come to the application. And one of the messages of today's talk is we really think that that's the time to go to the application, be it pan-genome or the next waves of large-scale genome comparison, or other applications that are interesting either for the breeding community or for the research community. So there's diversity, right? Maize is a very good example of it. A very important crop, huge diversity, needs to be managed. The usual way of understanding diversity is you take a lot of reads from different genomes and you try to align them to some kind of a reference that represent the diversity of the organism. However, it's difficult because short reads don't have a lot of information in them, and the practicity of aligning them is problematic in many cases. You would get some of the regions that are different in your genome versus the reference genome. Some region will be repeated, some region will be just very different, and so on. And basically, at the end, you're getting misalignments. And misalignment cause you to get not the outcome of the misalignment will get you a wrong polymorphism that is not very useful, and that limits your discovery ability. Okay? So what's the solution? The solution is to assemble. And one of the things that NRGene brings is the ability to assemble really quickly in high quality in a very cost-effective way, which makes it something that everybody can use from now on. So when you assemble, you filter out the noise. If you have repeats or heterozygosity, you phase it out, and you end up with something that is a long sequence. And you can think of it as computationally getting to long sequences instead of trying to manufacture long reads. That it's also a valid approach, but it's usually more costly. So these are data from different coverages of the same genome. If you do 180x, like we showed in many of the genomes that we presented throughout this PAG meeting, then you get to above 9 megabases of scaffolds, which is very good, very useful for a lot of the application downstream. The genome size is very big. This is maize. It's perfect, but it's expensive. How can you do something similar but be more cost effective? You can go down in the sequencing coverage, and you pay by the fact that the N50 is becoming shorter. But again, it all depends on the application. So this is still long enough to align it, for example. If you compare it to aligning short reads, it's still much, much better. And our vision is at some point doing 30x or 60x, and this vision is coming to a reality very quickly, is going to be so cheap that it's going to be a day-to-day practice in many of the labs. Now the question becomes, what do I do with all this data? And NRGene is coming with a solution. So just to understand what's possible today, this is the table of the last results from different assembly programs that were run with different customers or academic collaborations. You can see the coverages are around 200 and 180. This is Chinese Spring. So very long and complex genome, ending up with N50 of more than 7 megabase. And you can see we can handle a hexaploid, a tetraploid, different organism, even a fish. So it's really a kind of a universal tool. How do you translate this into genomic comparison or all the rest of the value that genomes can bring? So again, you want to be able to have the most out of the least coverage, the cost-effective way. So if you're doing 180x, you of course cover, this is a comparison based on a collection of very conserved genes that were published in a publication called BUSCO. Okay, it's a very conserved set of genes throughout the plantea, and it's more than 1,000 genes, and you can see that the blue part is representing the part of the gene set that is conserved. And 180x, this is the major part, of course, but even on the 30x, you can still get to around 90% of complete of the genes, okay? Which is almost what you need for most of the applications. If you drop that to a half, you start losing a lot of those complete genes, and you start seeing fragmented genes broken up between different scaffolds and so on. Okay, so just to prove, we can do diploid, tetraploid, hexaploid, if you're looking on gene content, and that's a usual measurement of the assembly quality, you're getting very high full gene content for all of those different weights. And the interesting part is that you can actually see the ploidity of the genome. So a diploid will mainly have one copy, a tetraploid will usually have 2 copies, and a hexaploid will usually have 3 copies. So that's a very nice result coming up of those 3 genomes. And again, you can see that comparing between gene content between genome can say something about what is this genome. So how practical is it to use? First of all, it's the time that it takes. So for the Chinese Spring, we got the sequencing done at about November 20, and by December 3rd, the assembly was done. So this is something that is now available for everybody, very quick, very cost effective, and can be done in large scales. It was done on a cloud machine, so you can do many of those in parallel. We're saying that the era has arrived to stop thinking about how do I assemble the genome, but what do I do with all those diversity? How do I handle the diversity? So the genomic assembly is becoming a technical issue that has been actually solved. Okay, so what do we do with all those genomes? The NRGene solution is called Pan-Genome, and it's part of a software package called GenoMagic Scholar, which we are kind of initializing in this meeting. And the idea is that we want to go from contigs and scaffolds to chromosomes, and then be able to do that in a very high fidelity manner, okay? I won't go into all the details, but I'm just pointing out that in different coverages, and you still want to keep a very high accuracy of mapping, you start to lose the ability to map part of the genome. And what you want to do is you want to balance the investment in specific genomes versus the utility of the information in those genomes for your specific cause. So if you want to have something in a very high fidelity, so you know everything about each position in this genome, you have to cover it in a high coverage and assemble it, but you can have a collection that also gives you a lot of information and saves you money. So this is kind of the idea of having many genomes. Each green point is a genome and comparing them all to all. And this is something that GenoMagic is now capable of doing. It's part of the GenoMagic Scholar version. So each one of the academic group that will actually license this software will actually have the ability to do this in large scale and in very complex genomes. Okay, what do you get out of it? So by comparing genome A to genome B through our mechanism, you'd first know the exact mapping. That is a very good starting point, right? You know where do you go from each position in genome A, how does it map to each position in genome B? But more important than that, you'd have the gene differences. So the CNV, PAV, any gene that has been duplicated or deleted, and the major translocation that happened between the genomes. And you can have that between each genomes in the collection. So now you start thinking like a picture of how my diversity is behaving from the mapping point of view and the gene content point of view. Okay. Usually, you want to go from genome complexity to some practical manner of doing something. For example, if you want to do breeding, or if you want to do SNP detection because you want to compare haplotypes between a different position in the genome. And the usual way you do it is you do SNPs. However, SNPs usually, the limitation of them is that they differentiate between a pair. So in this case, if you want to differentiate between PH207 and B73, you can use the SNP. The T is PH207, the A is B73. And this also looks like a good way to differentiate those genomes. Okay, those T's here and the G's here differentiate them. However, if you're starting to do multiple genomes, then the picture changes. And okay, and if you're just looking on those T nows, they're not useful to differentiate between this collection. They're only useful to differentiate that collection from B73. So what you want to do is you want to find those sequences or polymorphisms that are actually informative and can detect only the haplotype that you're looking for, okay? So you need to go through all the genome and understand which of the polymorphisms are actually identifying uniquely that specific sequence in be it B73, be it PH207, and so on. What we do is we use our assembly capability and the fact that we know the sequences to actually extract those markers, and we call them haplotype markers, and we take the full sequence, and now this little sequence here is becoming a marker for that haplotype. We don't use the SNP anymore, we use the full sequence as a marker for that haplotype. And the minute you do this, it becomes a universal marker for that haplotype because it appears only in this haplotype. That was the definition of it. So practically, we can now store SNPs, insertions, deletions, everything translates to a sequence, and you can extract that sequence from the assembly and store it. What do we practically do for maize, for example? We store those sequences in a database. The number of sequences we store for maize is about 20 million. And that's just collection of temperate maize. And only 25% of that number is SNPs. Okay? So we actually have a database that shows for each subsequence which line does it appear in. You can have thousands of those lines. And now we know if any new sequence come in, and that sequence will match a specific marker, we know that this is a marker for that haplotype. If you're doing GBS or any other things that translate into sequence, you can match or impute that specific sequence. So what's the value? Okay, you're creating a uniquely defined locus, okay, for each variant. And then you're using it to store all the different markers or the different polymorphisms that are there for a specific organism. And then you use it to cost effectively, and the point here is the cost effectively, being able to impute back. So we've created a connection between the GBS and a full sequence. You can impute back from those markers back to the full sequence. How do we actually use this? Okay, you have two genomes. Each genome has its own markers. And now, when you cross those genomes and you have a population, each individual in the population can be then, imputed back to his genome. Okay, so you go along the genome of the individual and you check what are the markers, and apparently, and it's like a step function. So when you're actually moving between a collection that matched genome A to a collection that matched genome B, it's a very sharp change, and you can actually detect exactly the points in which the change happens. It's very, very useful. And that's bringing us to mapping. So now you can genotype using those haplotype markers, and you then can do this collection of haplotype information. You usually collect a set of genomes in a very high coverage, and that's the base for your collection. You add some other lines that are relevant, but you don't need each in every point, so you're just covering them in kind of 20 to 30x. And then you use that collection to impute back from many, many GBS data sets that are the practical day-to-day use of the breeding or the research that you do. The other thing you can do with it is, of course, map. So you add the phenotype. You're correlating the phenotype to a specific position in your genotype collection, and in this case, this position with that haplotype is correlating with a specific phenotype, and the system then allows you to drill down and see the underlying sequences with all their gene content. So that QTL or GWAS analysis is actually application that we add to GenoMagic that will immediately be available for the academic customers of GenoMagic Scholar. So what is the system? How does it work? How does the GenoMagic Scholar comes to the customer? It's a cloud-based installed system that has a unique database that is originally, it was designed to be a big data database, and so it can grow and it can hold the information of a large community. And then you have this layer of API that allows people to start using it, and that usage can be, because it's a cloud-based, it can be kind of span over many geographies and in many places at once. And the usage is really depends on the need at a specific time. If you want to grow, you can grow in the cloud. If you don't want to use it, you shrink. The idea is that this is very matching how usually a community would work. There'll be people joining in, adding their data, using a research, maybe stop using it for a while, and then another part of the community will join. That's a very flexible model of sharing the IT resources and the database. So just to summarize, what I've shown you is that GenoMagic Scholar holds the ability to assemble very fast and quick. Then you compare genome to genome, you get a pan-genome that allows you to understand the genome, the mapping and the gene content. You get all this on a big data system that allows you to hold all the haplotypes, the haplotype markers, the mappings, and everything that you actually need to activate the system. And then there's an interface that will actually bring all that complexity to the user in a user-friendly way and allow the different users in the community to actually share this data and share all those expensive resources. So thank you, and I'll take some questions. Yeah, go ahead.
So for your assembly, do you require that paired end read data is prepared using TrueSeq protocol, or does traditional Illumina library prep works?
It's traditional. We have like a set of instructions for our customer. It needs to be PCR free, but
Right,
there's
so
nothing very unique about the paired end preparation.
And what are your requirements on paired end read lengths?
You usually use around 150. Some of the providers can actually get more of the machines and get to 160 in a kind of a magical way, which is really magic for me. But we don't need anything unique.
Okay, thank you.
Anybody else? Thank you so much.
Thanks, Gil. Thanks, Guy, for this opportunity. And as Gil mentioned, the W22 genome sequence is what Don McCarty affectionately calls a guerrilla genome sequencing project. So none of us had a budget or a grant to sequence the W22 genome, but we all had a need. And everyone that's listed up here has contributed in some way to the sequencing of the W22 genome. And this has all happened really in the past year. So I'm here as the representative, but it's been a really great group of folks to work with, including NRGene. And again, the driver for us was cost. We wanted to get the W22 genome sequence for a number of reasons. Personally, it was to map some of our transposons. We had struggled for a long time mapping flanking sequences from W22 onto a B73 reference genome because of the inherent polymorphisms between inbred lines of corn. And so why W22? So I'll start with a little bit of a history lesson for W22. So the name W comes from Wisconsin. This was a line that was developed, the Wisconsin Agricultural Experiment Station. And it was really pioneered by R.A. Brink and used in his very early studies of paramutation. And unlike many of the Midwest varieties where they had strong suppressors of color, Brink was able to use W22 and get good color expression. So we could look at things like the bronze locus, the R locus, the P locus, and they expressed well in this inbred. So it wasn't an inbred for high productivity, it was an inbred for maize geneticists. It was about this big, the tassel and the ear had good ASI, they were basically ready to be pollinated at the same time. It was easy to work with. And because of Brink's adoption of W22, he, his colleagues, and his students all also used W22. And so Oliver Nelson did some of his pioneering work on high lysine corn, looked at the bronze locus, was actually the first one with Nina Fedorov to tag a gene using transposon tagging, and looking at recombination in waxy1 with this W22 inbred. Jerry Kermickel's elegant studies of the R locus and complex rearrangements were performed with W22, and he also looked at DS transposition. And of course, Hugo Duner, who this conversation really started with, as many do at a maize meeting over a beer, said, you know, this would be a really great genome to sequence. And so because of these pioneers, there was a tremendous amount of genetic resource that had been developed in W22. And then later on, Don McCarty and his colleagues at University of Florida adopted W22 for their uniform mu project. So we have lots of transposon resources in this inbred. And that really was the start of it. And it's actually grown much beyond a simple genome sequence and assembly. But that's where I'm going to start. And this summary slide just shows you some of the stats that Guy just put up earlier. So you can see the scaffold N50 of 8.2 megabase is a contig, N50 of 32. This was the first iteration. We had about 4.5% gaps. The second version of the De Novo Magic software reduced our gaps to 1.8, and our scaffold N50 up to 9.4, and the contig N50 up to 73. And this was the number that I was most interested in for the placement of our transposons. So the transposons that jump into introns or into promoters, we want to be able to link those to the gene space. And we've been able to do that. So when we saw these numbers, we were quite impressed, but like probably most of you in this room, we're also a little skeptical. How could these guys sequence this genome so fast, put it together so well? And I still think part of that trick was the DNA quality. So Julia Verbalov, who's a scientist at Boyce Thompson Institute, isolated very high quality cesium chloride DNA, and that's what we provided to Alvaro for his library construction. Relatively straightforward libraries, mate pair libraries. None of the libraries were bigger than 8 KB, and yet they were able to put this genome together in relatively no time. And so Fei Liu in Ed Buckler's group did the first QC of the assembly. They had positioned about 4.5 million markers across the maize genome, and so they were able to see how well do the markers, in this case what I'm showing here is a region of chromosome 9, how well do those markers match the predictions of the Buckler lab. And you can see here that these lines don't do much crisscrossing. There's quite a bit of synteny here between the W22 and the reference map. So there are a few markers that bounce to other places in the genome, but at the time, I remember Ed saying he was really pretty amazed at how well this assembly looks. Another way of looking at this, the quality of the genome, was comparing a published back sequence that Hugo Duner's group had generated from W22 and comparing that to the NRGene Illumina assembly. And you can see we basically have a straight line. There's some repeat sequences that were hard to put together, but for the vast majority of the sequence here, we had really good alignments. And what this shows here is just a cartoon of the gene space, which is shown in black, retro elements shown in red, and then these missing annotation regions that are missing annotation. And this is very typical of the maize genome. So you have these large retro element Cs with these small little islands of gene-rich space. So Charles Dew dove in a little bit deeper, and he actually went in and manually curated a region around the Brown's locus. So again, he had published with Hugo a map of the Brown's locus using Sanger sequencing. They had really just pushed through this entire locus, and so they could see this series of nested transposon insertions. And so what he was able to do is take that region from the NRGene assembly, line it up to this back sequence, and manually curate. And so for whenever he saw a gene, these are the hits, these are the percent identity. So this hypothetical protein 4 was 100% identical to the W22 back sequence. And again, you can see these regions here of 99 to 100% identity. Where it started to break down was in these big retroelement clusters. So he knew from his sequencing he had an 85 KB cluster. In the NRGene assembly, about 6 KB of that 85 was actually missing, and then there was about 1 KB that was inserted that shouldn't be here. So what this suggests is that with the short read sequences, it's hard to really push through these complex rearrangements, but when we get into gene space, and even the intervening gene space, as long as we don't run into these sort of nightmares from a sequencing perspective, it's possible to do a very good alignment. So again, as Guy had mentioned, another way of assessing the genome quality is through these BUSCO scores. So it's a benchmarking of the universal single copy orthologs. So this is really just looking at genes that are conserved across all organisms, or in this case, what we looked at is the set of BUSCOs that were present in all plants. So you can think of these as sort of a core set of genes that define plant genomes. So the idea is that If your quality of the assembly looks good, then you should have a high number of these BUSCOs in your genome. And so the W22 we compared to version 3 of B73. So when we looked at the single copy BUSCOs, there were 908 in W22 compared to 894, so very comparable quality of complete BUSCOs. Maize is an ancient tetraploid, so you can see there's some duplicated ones in maize, and those similar numbers are again very similar to B73. And we actually saw fewer fragmented BUSCOs than the B73 version 3, and then the ones that are missing is only 32. So that's about 3% of the conserved gene space that's missing. So another way of looking at that is we've captured probably about 97% of the W22 genome with this assembly. Okay, so with those quality assessments done, we then worked with Yinping in Doreen Ware's lab. Sorry, keep doing that. And so what Yinping Zhao has done is run the W22 genome through her Maker-P pipeline. She did a repeat masking and then F genes H ab initio calling, and then used RNA-seq data from 8 different tissues to polish the gene models that she has from the ab initio pipeline into about 70,000 full-length cDNA, and included 70,000 full-length cDNAs from B73 and Pioneer. She also added some genes from other species and to derive a set of what we call 57,000 working gene set, which is comparable to the working gene set of B73. This was filtered further into a high confidence gene set, and that number here is about 40,789 genes, which is very similar to the number that was derived at B73. So from these metrics, it looks like the quality of the W22 genome is as high, if not higher, than the current B73 genome. Brad Barbazuk and Wenbin Mei in his lab then went a step further and characterized alternative splicing events in this genome. Again, they used a large collection of RNA-seq data that was sort of cobbled together from all of the collaborators. And what they found is that roughly 34% of the genes in the W22 genome are showing some form of alternative splicing, whether it's alternative splice acceptors, donors, terminal exon variants, or exon skipping and intron retention events. And so they've represented those sequences for viewing in a number of different browsers, and those will be accessible to the community. Daniel Vera in Hank Bass's lab has really been the go-to guy for organizing and assembling all of these diverse data sets, and to set up a number of browser shots. In this case, he's using this UCSC browser where he stacked the gene expression levels. So each of these different colors represents gene expression from a different tissue, and it's being mapped back onto the genome. So you can quickly scan through, look to see where your favorite gene is expressed in the appropriate levels in the appropriate tissues. Carson Andorf and John Portward have done a similar thing with Maize GDB. And so now, again, we've got the W22 genome incorporated into Maize GDB. You can look at gene expression as different tracks shown here relative to the gene models. Okay, and so as I mentioned at the beginning, one of the reasons we did this initially to begin with was to have a better placement of our transposons. Dal McCarty, Mark Settles, Karen Koch, we're all interested in where mutator lands. My group, Eric Volbrecht, and Hugo Duner's group, we're also interested in where all of the DS insertions mapped. And so we can see here in these pie charts is that for the vast majority of genes, there's a good conservation in the map position of the mutators and the DS elements to the B73 genome. So all of our placements had been based on flanking DNA. We aligned it to that reference genome, and about 84-85% of those matches seemed to be conserved with B73. However, we identified 16 what we're calling novel placements with mutator and 12% with DS. And that translates for mutator about 4,000 mutator elements that we were unable to place in B73 that now find a home in W22. And that the ability for us to be able to do that probably represents presence-absence variation or significant polymorphism between W22 and B73. So we've increased the value of this resource by being able to place more of those elements. We also saw a number of elements that we call relocated. So they were originally placed on one chromosome in B73 and now fall on a different chromosome with higher accuracy in W22. The numbers we had for DS are smaller, but again, still significant. So we were able to position 207 DS insertions to novel regions within the W22 genome, and 38 of those were relocated. Another really slick analysis that Hank and Daniel led was what's shown here. So they performed both light and heavy digests with a micrococcal nuclease. And this micrococcal nuclease sensitivity is a way of monitoring the open chromatin sites. So the way to visualize this is the blue here is showing regions of the genome that are accessible with a light digestion of the micrococcal nucleus relative to a heavy digestion, which is shown in red. And these light regions are associated with open chromatin, and these are associated with nucleosomes. And what we can see here is that when Daniel mapped the mutator positions relative to these micrococcal nuclease sensitivity sites, you can see a peak right at the mu insertion site. And we do not see that same bias with DS. So that suggests that the mutator elements are looking for these open chromatin structures. And we do see a number of mu insertions when we look globally across the genome that fall into promoters or UTRs that would be consistent with this micrococcal nuclease mapping. And again, this was performed globally, and all of this data will be visible and visualized on the UCSC browser or on Maze GDB. Nathan Springer also did bisulfite sequence analysis to look at the methylated GC content. Again, found what we had known for a long time through restriction analysis is that DS, mu, and AC all like hypomethylated regions of the genome, so they tend to land very close to low methyl regions. But what was interesting is that mutators show that mutator elements tend to sit very close to the borders of both high and low methyl CHH islands. So again, another structural feature of the genome that mutator likes to insert near. And so again, with these results, we can start to potentially develop some machine learning algorithms. The hope is that we'd be able to do a better job of predicting where some of these transposon insertions lie. And we don't see that same bias with DS. So, this, in summary, the comparisons of these mutator and DS footprints suggest that mu and DS are likely to be very complementary transposons in the sense that they're seeking different signatures of the genome. They're looking for different heterochromatic marks within that genome. And so I guess in the final couple minutes, I'd like to conclude with how this improved mapping of our DS transposons is actually helping us drive some biology. And so members of my group are part of a large consortium to look at installing nitrogen fixation in the grasses. This is a project run by Giles Oldroyd and supported by the Gates Foundation. And our role in this project was to identify a number of potential components of a mycorrhizal signaling pathway, disrupt those in maize as a starting point for genetic engineering. And so we decided to use the DS transposons in W22 so that we would have a uniform genetic background to do some of this detailed characterization. So we identified DS insertions upstream of those 1, 2, 3, 4 genes that I have highlighted here. And what's important for using DS in mutagenesis is that we often don't have the transposon in the gene of interest, but near the gene of interest. And the frequency of insertion into the target gene is directly correlated to the proximity of that DS to the target gene. So accurate placements in the genome is critical for DS tagging. And so we had a DS in this case that was originally in a B73 genome, approximately 300 kb away from the target gene. And in our sort of testing of this system, 200 is usually the limit the cutoff that we have for using DS. So if a gene is more than 200 kb away from the transposon, we don't feel it's a good target. It's too far. So this was a DS that was a bad source, but then when we had the W22 genome, we realized it was actually only 57 kb away. So Twin Zhang, who's a postdoc in the lab, created a population of 3,000 individuals, and then with PCR basically mapped 4 independent insertions into this gene, and is now effectively has mutagenized this gene and is doing a more detailed characterization. So I put this up here as just one example of how that improved mapping in the W22 is being exploited to do novel biology. And again, because all of these transposon resources have been developed in W22, I think it's going to be a tremendous resource for the community. And so with that, I think I'll end it, and I'd be happy to take any questions.
Hey, Tom. That was great. You and the previous speaker said it was very cost effective to do this. Is there a number that you could quote for, I give you DNA and you give me back an assembly of this quality?
Well, you know you're going to have to wheel and deal with Guy and Gil on that one. So
let's do it. Let's do it.
So, yeah, for us, none of us had a grant. It was all limited by our budgets, and so the cost for us, sequencing, assembly, the whole 9 yards, was $50,000. And this was about a year and a bit ago. The sequencing was probably half of that cost, and we really couldn't do it cheaper. We certainly couldn't have done it better at that time.
Great, thanks.
Sure.
Good evening. My name is Guangtu Gao. I work with USDA National Center for Cool and Cold Water Aquaculture. I'm glad to talk our research project and share our experience on genome assembly here. This presentation is about the genome assembly for rainbow trout. To give you some background information about why we want to make a rainbow trout genome assembly, I want to show you first about the mission of the National Center of Cool and Cold Water Aquaculture. So you can see from this text, our majority of our research project related with rainbow trout. So it's very important we have a rainbow trout genome assembly in a good quality. So that's why we want to do this project. So before our work, there is a rainbow trout genome assembly developed by the scientists from INRAN, majority from INRAN. And this paper published in Nature in 2014. And this is what we call the current version of rainbow child genome assembly. And this assembly, when we use it, we feel, it did a good job in gene discovery and annotation. However, we also found that the current version of rainbow child genome assembly, the N50 is not quite satisfactory, and also a lot of scaffolds are not anchored or other in a chromosome. So the chromosome sequence from these assemblies are not quite useful. So that's why we want to do a new rainbow child genome assembly. This work is still ongoing. It's not finished. So today I'm just sharing some initial result and also to share with you, what we plan to do after we get the data from NRGene. So I'm going to first talk the assembly, and then I will talk about something about this, how we plan to use the linkage map to improve the assembly, and also how we use the linkage data to make the chromosome sequence. And at the end, I want to compare the new assembly, some initial data of the new assembly with the current version of rainbow child genome assembly. So we start this assembly by sequencing the rainbow child double haploid Swanson line fish. The same sample was also used by the current version of rainbow child assembly. And we basically, the sequence were generated from 5 libraries, 1 parent library and 4 mate pair library. So we got a lot of sequence. This is the coverage. And those sequences were done by the sequencing center in University of Illinois. And then those sequence data was sent to NRGene, and they used the pipeline, their pipeline, de novo magic, and made the assembly. And those are the data showed the result of the assembly. So one notable data I want to show here is the N50 of the scaffolds. So this assembly with NRGene is 1.7 million, over 1.7 million base pair for the N50 for the scaffolds. And the current version genome assembly is only 383K base pair. So we think one of our goals is achieved with this assembly. That is we hope to increase the N50 of the scaffolds. So after we got all these assembly results from NRGene, so we need to do some bioinformatic process to those data and to make the chromosome sequence. So we decide first we want to use the linkage data to improve the assembly and to generate the chromosome sequence. This is a pipeline we adopt from the Atlantic Salmon Genome Project. And we first discovered the SNPs in rainbow chart genome, and then make the linkage map for those markers. And then using those mapped marker, we try to find whether there's arrows, scaffolds in this assembly, and then we break those one. If we make sure, this is really some problem, so we just try to find a place to break the scaffolds, and then we put all these scaffolds in the same chromosome, and using different methods, try to make a super scaffold, and then build the chromosome sequence. These are the pipelines we are doing for improving the assembly. The last two, I would say, this is ongoing. The last two steps are not finished. We are finished the first three. The markers we used are from three sources. One is the rainbow chart SNP chip markers. There are around 57,000 SNP markers in the rainbow chart SNP chip, and we use this SNP chip genotype many fish, some from the Norwegian rainbow trout population, some are from the USDA's breeding populations. So using this genotype data, we can put about 48,000 SNP marker in the linkage map, and this is the SNP chip marker that can be linked and in the linkage map. And we also use two other source SNP markers from the rat sequencing technology. And one group of the marker are also in the linkage map, and the other group are, the markers are in the linkage group, but not ordered in the linkage group. Basically, all these three groups of marker, we can use this marker and map to the new genome assembly, and over 77,000 markers can be found in the genome. And those markers are in 3,293 scaffolds. So it's about those scaffolds takes about 82% of assembly. And among these number of the scaffolds, 3,214 of the scaffolds are uniquely mapped to one chromosome. That means the linkage map is consistent with the assembly. That's a good news. But there's still 79 scaffolds. They are mapped to multiple chromosomes. There are many, source of reasons for this situation. It's not just because of the the assembly was wrong, and so we make some requirement to find the problem scaffolds. So these are what we required to do this scaffold breaking process. At the end, we found 33 can be, we are sure we can break. And then that's the length of each chromosome after we arrange the scaffold into the chromosome. So at the end, we can. So after we got the scaffold, the chromosome sequence, the ongoing project is, we want to join those chromosome sequence into super scaffolds, and then make the annotation. So after that, we can publish this new rainbow chart genome assembly and use it for the research project. So this table just show, at the end, I want to show the comparison of the new rainbow chart genome assembly with the current rainbow chart genome assembly. So we can see that the size of the assembly is increased a little bit, and the scaffold map to the multiple chromosome, the current one is 79. After the breaking is, we also broke 36 scaffolds, and then the current version of a rainbow chart genome assembly is, there are 330 scaffolds are mapped to the multiple chromosome. So that means the accuracy increased a little bit also, and also the percentage of anchored base, the current one is increased from 48 to the new rainbow chart genome assembly is increased from 48% to 82%, and the percentage of altered the bases increased from 5% to 82%. That means we have a much better chromosome sequence with the current genome assembly. So the take-home message is that the new rainbow chart genome assembly is nearly done. The first version of the new rainbow chart genome assembly is almost done. So it's a great improvement over the current genome assembly. That's the message I want to share with you. At the end, we want to thank our project, our funding from both the competitive grant and also the in-house grant from USDA. And this project, there are many people contribute to this project. This is the list of the contributors. That's the end of my talk. I would like to answer any question regarding this data. All right.
Thank you for the introduction. I will speak about the quality evaluations that we did with the NRGene assembly of hexaploid wheat, and then with the about anchoring and ordering the scaffolds to get a chromosome scale assembly. And for this we used chromosome conformation sequencing data, so Hi-C. So first, some introductory words about wheat. So wheat is an important crop, and as you may know, it's a polyploid. It's a hexaploid. It has 3 subgenomes. These are the A, B, and D subgenomes. So wheat arose from 3 rounds of genome duplications from allopolyploidization. So in the first round, the A and the B genome were created to merge to create the AB wheat, or Triticum turgidum. This still exists. This is durum wheat that is used for making pasta, and probably after domestication or shortly before domestication, the durum or emmer wheat merged with a third species that is called Aegilops tauschii, and this created what is nowadays bread wheat, so a hexaploid species. But actually, it's a benign polyploid species because the subgenomes are well diverged from each other, so there's about 3 to 4% divergence in genic regions. But still, wheat is has a very large genome. The genome size is around 15 to 17 gigabases, and it is highly repetitive. About 80% of the genome is repetitive, and but the gene content is highly conserved between the subgenomes, so the gene order along the 7 chromosomes is conserved, and the chromosome number in the subgenomes is identical. So because of this large complexity of the genome, it was long thought that you need a back-by-back approach, so hierarchical shotgun sequencing, and this was the approach that was followed by the International Wheat Genome Sequencing Consortium for 10 years. So to speed up the process, in the meantime, the consortium IWGAC created a chromosome shotgun sequence where they sorted individual chromosomes and assembled them from shotgun reads, and this was published in Science in 2014. And then last year I reported about the whole genome shotgun assembly that JGI did also of hexaploid wheat, and this established that it is possible to get good whole genome shotgun assemblies. And now IWGAC has also created a whole genome shotgun assembly together with NRGene. And the statistics of this assembly are really impressive. So in this plot, the closer the line is to the left, to the upper left corner, the better the assembly statistics are. And you can see that the NRGene assembly is 2 orders of magnitude better than the published assemblies, and also better than another whole genome shotgun assembly that was recently released by PAG. So we were very impressed by these statistics, and of course we had to do some validations because we could not believe these numbers when we first saw them. And actually, NRGene did not do only 1 assembly of wheat, but 2 assemblies. So they also assembled Emmer, the wild Emmer genome from Israel. So Emmer is a tetraploid wheat. And as you can see, the genome sizes, the assembly sizes come very close to the estimated genome sizes. The N50 are in the megabase range, so around 7 MB for both species. And so 90% of the assembly can be covered with contigs that are larger than 1.2 MB, and around 2,000 contigs can cover 90% of the genome. Also, we used high density genetic maps with millions of markers to anchor the assemblies, and because of the large contig size, most of the assembly could be assigned to chromosomes and anchored genetically along the chromosomes, so over 95% of the scaffolds have genetic positions. This genetic data also showed that there are some inconsistencies in the scaffold, so where there are wrong contig joints. So for example, in this assembly we have a 28 MB scaffold that joins 2 or more contigs from different chromosomes, so 1 from 5B and 1 from 4D. And we provided this information to NRGene, and I think within a couple of days they could correct these chimeric scaffolds and break them up and produce an updated version of the assembly. So it clearly shows the advantage of having a high-density genetic map to correct these assemblies. As another step of validation, we took a finished bug-based reference sequence of 1 wheat chromosome of chromosome 3B that was published in Science in 2014, and we looked at all the scaffolds, and this plot show the scaffolds of the 3B reference sequence, and these plots shows the alignment of the 2 largest 3B scaffolds to the NRGene scaffolds, and you can see that the complete sequence of these bug-based scaffolds is also present in the NRGene assembly with perfect collinearity, except for here we have an inversion, but we don't know which of the assemblies is wrong here. What is most important is that the NRGene assembly captures all the sequence that is also present in the backs, and it has an even better contiguity. So we have a 4 MB scaffold in the 3B reference that is contained in a scaffold that is 28 or 22 MB in the NRGene assembly. We also did a comparison to the physical maps. So for most of the wheat chromosomes, there were physical maps that were constructed by high information content fingerprinting or by the whole genome profiling method of KeyGene. And here we have a single NRGene scaffold that is almost 29 MB on chromosome 6A. And we can align over a dozen contigs of the physical map to the scaffold, and we see perfect collinearity. And so the scaffolds that NRGene produces are even better than a physical map. They have a better contiguity, and this information also allows us to assign back sequences to scaffolds. So it's not necessary anymore to really sequence minimum tiling paths of the physical map, but we can just use the available scarce sequence information or survey sequence information of the physical map and integrate the back sequences with the NRGene assembly. So then, for the rest of my talk, I will speak about the chromosome conformation capture, or Hi-C, that we use to order the scaffolds along the chromosome and get chromosome scale assemblies or pseudomolecules of the wheat genome. So Hi-C, you heard about it today in the plenary lecture. It's actually a method to query the 3D conformation of chromatin in the nucleus, and this describes the wet lab protocol, which is a fairly advanced protocol. First, the DNA or the chromatin in the nucleus is cross-linked in with formaldehyde, and then it's cut by a restriction enzyme. The restriction ends are filled in with labeled nucleotides, and then they are religated. And so during the fixation step, DNA molecules that are close by in the nucleus in the 3D structure are close to each other, and they are cross-linked. And then during the ligation, these chimeric molecules are formed that contain DNA sequences from different locations in the genome. And these chimeric molecules are then sequenced on the Illumina platform, and you get the two ends of the read correspond to different genomic locations. So you can now look at all pairs of genomic locations. So for example, in 1 MB bins, and count the number of Hi-C links or chimeric pairs that you get that link these loci. And what was established in humans is that you have something that's called the distance-dependent decay. So the farther apart two loci are in the linear DNA sequence, the farther apart they are also in 3D space. So there's a high correlation between distance and linear distance and three-dimensional distance. And this inspired people in the human world to use this 3D distance that comes out of Hi-C to use this as a proxy for linear distance, and use these linear distance to make a map of the linear chromosomes. And they made a proof of principle experiment where they recreated the order of scaffolds in the human genome. So they used publicly available Hi-C data to make a map of the human genome, and they established that really the 3D conformation can be used as a proxy for the linear order of scaffolds on the chromosome. So we also made Hi-C libraries both for bread wheat and for emmer, and used this to order and anchor the scaffolds of both assemblies. So we get several million Hi-C links, and these links, so these are links between scaffolds that are really informative to make the map. And by the high-C mapping procedure, we could anchor over 96% of the assembly in both cases. So, and anchored or ordered means really that we have a fully resolved linear order along the chromosome. And with the higher density high-C mapping data in wild EMMA, we also could orient the scaffolds. We know which end is closer to the telomere or to the centromere. And we could anchor also over 90% of the, or orient over 90% of the EMMA assembly. So on average, each chromosome is represented by less than 200 scaffolds now. So for comparison, the previously available assembly of bread wheat of the reference assembly Chinese Spring had over 2 million contigs, or several thousands of contigs per chromosome. Now we did a validation of our high-C map because it's quite a new method, and we were not sure if it really produces correct results. And this shows the comparison of the high-C map to various other resources. So the first one is a KARMA. This means chromosome arm assignment. So it's possible in wheat to sort individual chromosome arms and get sequences only of the chromosome arms. And with the high-C data, we can see a clear break between the short and the long arm. So it's possible to actually map the location of the physical centromere with the high-C data. The next plot is a comparison to the POP-seq map. So POP-seq stands for population sequencing. It's a method to construct an ultra-dense genetic map by sequencing the members of a biparental mapping population. And we see perfect collinearity, but this vertical line corresponds to a well-known problem in wheat genetics, that we have large genetic centromeres where there is no recombination. And so we have a single bin that extends to over half of the chromosome, but with the high-C map, for the first time, we could resolve this non-recombining paracentromeric regions. And really, the best proof that we are really correct is a comparison of the subgenomes in wheat and bread wheat. So this shows the comparison of chromosome 2b, first in the Chinese Spring, so the bread wheat reference variety, compared to the 2b in the tetraploid wild EMMA. So the genomes should be highly collinear, and this is really the case. We see collinearity throughout the genome, and we see a little bit of breakdown towards the centromere. And this is a trend that we will also see later in interspecific comparisons. What is also interesting now is that what we can do with this data, we can look at translocations or regions of suppressed recombination. So this is an example for chromosome 4a. We see here the pericentromeric region that we expect on each chromosome, but then we see another also very large region where we don't see any recombination, where we have a single genetic bin that is resolved in the high-C map. And on the right-hand panel, we see the most likely reason why there is no recombination, because we have a large inversion between the 4a in bread wheat and the 4a in EMMA, and this inversion is responsible for the suppressed recombination. So with the high-C maps, with the high-C maps both of bread wheat and EMMA, it's now possible to do comparative genomics to look into structural genome variation at a phylogenetic scale. We can also compare different species because we're within the framework of the Barley genome sequencing consortium. We produced also a reference sequence of barley and also made a high-C map of barley. And now we can compare the maps of EMMA, bread, wheat, and barley. And this is shown here for one chromosome, where we compare the chromosome 1A of EMMA with the chromosome 1 of barley. And we expect a good collinearity, and we see this, that we have overall collinearity, but we see these inversions here. And interestingly, these inversions here on the long arm, they are conserved between the 1A, and so between 1A and 1B, so between both subgenomes, so probably they arose before the A and the B genome diverged, but after the divergence of wheat from barley. By contrast, this inversion here is only present in 1A and not in 1B, so this is maybe private to the A genome. What is also an interesting pattern is this breakdown of collinearity in the pericentromeric regions, and this is consistent with the notion that centromeric regions are a driver of speciation. Yeah, with this I'm at the end of my talk, and I want to thank first the bread wheat assembly team that was led by Nils Stein, Curtis Postniak, and Jesse Poland, and was supported by the International Wheat Genome Sequencing Consortium. And I want to particularly mention Axel Himmelbach, who established the High-C protocol and did the wet lab work at IPK Gattersleben. And the other half of the work was done by the Wild Emma Wheat Sequencing Consortium that was led by Asaf Distelfeld. And yeah, I thank him for let us join this consortium and really have a look at these NRGene assemblies. And I thank you for the attention.
This is the project that I keep hearing impressive information from the maize community. And I basically, well, in the soybean community, we have a glycine max domesticated soybean reference genome, William 82. It was published about 5 years ago. It had always been my aspiration to do a reference genome for wild soybean. And we don't really have a lot of funding to do it. And I basically say, well, let's try NRGene with all of the good information that I've been hearing about. So we were able to work out a collaboration with NRGene. And it is really fast and impressive and high quality assembly. I can testify to that. We just got the data. We actually sent them the DNA basically around December the 10th. And by the end of the year, we got the assembly, and we spent a couple of days with the team in Israel to go over some preliminary analysis, and it is impressive from our perspective. So with that, I guess I'm going to ask Babu to give you some information about the wild reference genome. Thank you.
Thanks, Andrew. Yeah, they're right. I mean, like we got this kind of yesterday kind of stuff. So we sent the DNA last month, and we got the data this month, and we started to put something together. So we are giving just a highlight of the assembly. So before that, I'll give you a brief introduction about soybean and why we selected this line. So you see that this is the year of legumes. It plays essential role in food security. And this one. Okay. So soybean is one of the cash crop in the US and all over the world, and mainly used for it's tricky. Food and feed and energy. Then we all know that we had to make the crop production double by 2050, so we had to work for that. Then for that, the major problem with the soybean is it's reduced genetic diversity, so we have to pose a lot of challenges to improve yield. So for that, we had to look into the evolutionary aspect of the soybean, and it originated from a common ancestor. And soybean originated, the cultivated one originated from the wild glycine soja. It's about 9,000 years back. And you can see the difference between cultivated and wild species. The wild species have distinct characteristics when compared to the cultivated one. And most of the wild species are originated from the East Asia and some part of the eastern part of the Russia. So why we focus on wild? Because one crop strain cannot give you complete history of the gene or genome evolution. And also, these wild species have a potential resistance genes and other agronomic important genes. So use that untapped resources for a crop improvement program. So we're looking at the genome sequencing of the legumes. So the glycine genome was the #1 in 2010, as Henry mentioned, then followed by the other genomes in the legumes. So the first cultivated soybean genome, Williams 82, is about 1 gig, and we assembled about 978 MB. And along with after the cultivated soybean, there are some efforts from the wild soybean genome sequencing folks. One from Sukhalis in Korea, that's the first one they published. Then from Hongming Lam and BGI, published did about 17. And recently, Li Chu from China made a pan genome for about 6 or 7 wild species. So in summary, it's a modern cultivated soybean is from diversified from about 5,000 years back, and G soja is from a broad geographical range, and we can have much genetic diversity. So same approach people did in other crops also, like solanum till other important crops. So we selected this PI, this plant introduction 483, 463 for genome sequencing because it's having some specific characteristics. First of all, this is kind of most diverse soja from our soja collection in the United States, germplasm, and it's having a biotic stress tolerance, and we have the maps available. And it's the important thing is this: it comes under the maturity group 3. So we can make crosses the part where we are from, and it's well characterized for other stresses and compositions. So project goal: assemble, annotate, make a reference genome for soybean, and integrate all the sequences, especially genetic and physical maps, transcriptome data, and mapping data to improve the assembly and give this better assembly to soybean community. Because at present there is no complete reference genome for a wild soybean available in the world. So as Henry mentioned, we sent the DNA in December, mid-December to Illinois, and we did. Alvaro did a pretty good sequencing over there with different libraries. The first library is with a 400 base pair with the rapid V2 chemistry. Then second library about 700 to 800 base pair with there's Illumina V4 chemistry. The other 3 libraries using V4 chemistry about 160 base pair each. So total coverage is about 250x. So the genome assembly statistics, like this is the very prelim data what we got. We got about N50 is about 46.8, and the assembly size, we assemble pretty much of the genome, about 997, because it seems that it's a little bit bigger than the cultivated genome size, assembled genome size we got. It's about 11,000 scaffolds with the N50 about 46.8. And if you look at the contig assembly statistics, the total contigs is about close to 100,000 contigs we got. Assembly size with the contigs come to 962 MB, and the GC content, sorry, the N50 is about 23.8, and the number of sequence about 1,200. So so this is about assembly per chromosome. You can see better chromosome assembly. From the preliminary, we can see some better chromosome assembly stat here when compared to the William 82. And some of the chromosomes kind of shows a little bit bigger than bigger than what we got in Williams 82. So we compared this with Williams 82 available genome. The total scaffold Williams 82 is about 1,200. And we got, in the Soja, we got about 11,000. So assembly size about 978 MB. And in our case, we got about 997 MB. It's bigger. And gap size in max, cultivated is about 2.4%. And the Williams, in the wild, it's 3.5% gap. And N50, that's pretty impressive. The shotgun sequence what we used to make the Gmax is about 48.6, and with this Illumina-seq of 250x, and with this de novo magic we got 46.8 MB N50. So GC content, the preliminary analysis shows that we got about a little bit higher GC content in the wild species. So also they compared, they verified the genome using the recently published method, the Busco genes. So you can see the comparison between the available Williams 82 and the wild soybean about the using this Busco gene analysis. So we can see pretty much a very good excellent assembly on par with the available Williams 82 genome. With this, what we have a lot of things to do because we got this data only about, like he mentioned, Jill mentioned from the plane. So we had to anchor markers and maps and chromosomes and assign chromosome completely, and we had to complete the assembly and with annotation and utilize all the the wild soybean genome sequence information out and improve the assembly. And also utilize the transcriptome and metabolite data to make the annotation more meaningful. With this, I thank NRGene for their excellent help and collaboration and to get the first wild soybean genome, and also my lab members who helped in some of the process. Thank you all, and questions.